In the 1990s, an allergy drug sold around the world made some patients suddenly collapse, their hearts stopping. Strangely, the killer wasn't the allergy. It was a potassium channel inside the heart. Its name is hERG. Today, every new drug in the world must clear one gate before approval: hERG. Fail it, and billions in research can go to zero. It's called one of the valleys of death in drug development. What exactly does this gate test? And why can a single potassium channel decide life or death? First, how the heart beats. With every beat, a heart muscle cell first depolarizes, as sodium rushes in and the voltage flips positive. Then it must repolarize, pushing potassium back out and resetting the voltage, ready for the next beat. The main force behind repolarization is the IKR current, carried by the hERG channel. Here's the problem. Many drugs with completely different structures accidentally block hERG. Potassium can't get out, repolarization slows, the action potential stretches, and on the ECG this shows up as a prolonged QT interval. Once QT gets too long, the heart can fall into a lethal rhythm called torsades de pointes, then ventricular fibrillation, and sudden death. Back to that drug from the start, terfenadine. It does block hERG, but normally the liver quickly breaks it down into a safe product, so nothing happens. But once a patient also takes ketoconazole or erythromycin, or even just drinks grapefruit juice, that metabolism is blocked. The original drug piles up in the blood, and hERG gets jammed shut. So a safe allergy drug became a deadly one. In the late 1990s, it was pulled from the market. Cisapride and estemizole fell to the very same channel. That's why hERG became a mandatory test for every new drug, written into the ICH-S7B and E14 guidelines. How do you test it? Patch clamp is the gold standard. On a cell expressing hERG, a glass microelectrode seals the cell and records the hERG current. You add the test drug and watch how much the current drops, giving the half-inhibitory concentration, IC50. Then you compare IC50 to the drug's blood concentration. The safety window usually needs to be more than 30-fold to count as safe. Automated patch clamp does high-throughput screening, and manual patch clamp does precise confirmation. And today, testing hERG alone is no longer enough. The new CiPA paradigm evaluates a whole set of cardiac ion channels at once, plus heart muscle cells induced from human stem cells, to see cardiac safety more completely. From the tragedy of a single allergy pill to an entire suite of precise electrophysiology, this is patch clamp, standing as the last gate between every new drug and the human heart. Next time, why testing hERG alone is no longer enough. Follow Ion Channel Observatory, and we'll explain every channel that decides life and death, one at a time.